And thank you so much, everyone, for joining today's webinar. As Ashley mentioned, my name is Ghader Manafiazar, and for today,
pay attention.
For today, after I introduce and give some background information about myself, I will talk about implication of genetics and genomics in beef and somehow in the dairy cattle, and also what lessons can we learn to implement this technology in the lamb industry or sheep industry. So then, before that, I would like to answer two questions so that I have a better idea about the audience, like whether you are a producer or researcher and how much do you know about the genetics and genomics. Basically, knowledgeable, fair, little bit. And I didn't put intentionally zero knowledge because we know that genetics is a buzzword and we know that it's going to affect our life in a different way. So then I would appreciate if you answer the first two questions. We'll give about 30 seconds. Okay. And good. So then, actually, I'm not sure how to end the poll. And so I see that like a majority are producer and kind of the different knowledgeable. Like I see like the one person in the knowledgeable probably who's the researcher, and the others like the fair or little bit information. So then at the end of this seminar, I'm hoping that at least you get some sort of a general idea about the genetics and genomics and how you can implement this in your operation.
Just to confirm, do you want me to be closing the polling?
Yes, please.
Okay.
Yeah.
There you go.
Yeah. Okay. So about myself, I am assistant professor at Dalhousie University since last year, but I haven't moved to Nova Scotia last year and just recently moved to Nova Scotia. And today I'm joining you from Halifax, and it's a good region in case if you would like to visit. Really, it's a nice place. Before moving here, I worked at the University of Alberta in different capacities for about 12 years. Did my PhD and postdoctoral research associate, but my research interest is mainly about feed efficiency, greenhouse gas emission, fitness trait, reproductive performance. And I call myself as a livestock management specialist who has different skills, including genetics, genomics, nutrition, and currently I'm interested to implement sensors in livestock industry and do the machine learning projects. But besides my academic role, I actively engage with the beef industry and dairy industry. For example, because it's more related, I provide consultation and do the data analysis for Beef Booster Company. In case if you don't know, Beef Booster Company is based in Calgary, and they are active in the beef industry, and we sell crossbred bulls based on their genetics and genomic value to the Western Canada. So then I am the one who is doing the genetic analysis and give the consultation for Beef Booster Company. So then I sit by breeders, and we have 7 breeders. So then I sit with breeders and I talk to them and understand their concern and their objectives. So then we navigate their breeding decision. But I don't have just the experience with the beef. I worked with sheep for about 3.5 years. I was managing a research station back in Iran with about like 200 eaves. So then during my master program, we were interested to see the effect of crossbreeding in order to reduce the fat deposition in sheep. So then as you see in picture, like in Iran, we have sheep, we call that fat tail. So then these animals, they reserve fat around their tail. So then during the spring and summertime. So then later on in the fall or winter, they can release energy from their fat reserve in order to support their body function. So then, but you know, like it's not favorable trait from the fattening perspective, because you want to have a more carcass weight rather than the fat weight when you are fattening animals. So then we were interested to see that whether with the crossbreeding can we reduce the fat deposition or not. So then it was for 3.5 years, and we did the carcass evaluation. So then we found that no, we cannot reduce the fat deposition by crossbreeding, rather than we can reduce the fat deposition by within-bred selection. So then maybe some of this concept is not familiar for you, but you will learn more about this, the selection concept and others as we go forward. So then this is a really good paper, and it's highly appreciated by scientist community and industry. In case if you are interested, just you can contact me or Ashley, then I can provide the full manuscript, full text manuscript. So then that was about myself, what I do, and about my sheep background. So then I am kind of familiar with the concept of the industry, but to be honest, between the last 12 years, I haven't involved that much with sheep industry, particularly in Alberta. But the skill sets that I do have, it's a kind of the similar for any other livestock. For this presentation, I will walk you through some sort of the basic concepts for genomics and genetics, and why pedigree is important, and what are the advantages of genomic information, and also I will provide some examples. So then the question that I have, and Ashley, please bring out the polls, like how similar are we to our dad, or how likely we share genes with our parents, like it's a 50% or 25% or 100% or 0%. And the same case for mom. I appreciate if you respond this question. Give your best guess. The question is, what percentage of our genes are shared or are similar with our dad and mom? So then I will give like the 20 more seconds, then we will close the poll. Okay, so then I see like the response is 50%, and I'm ending the poll. Yes, we share 50% of our genes with our dad and mom. Indeed, mom has a little bit more contribution than 50%, but for the simplicity, we say, okay, 50%. And this is the basic structure to use the genomic advantage. So then we know that if we select good animal with a good genetic potential, then most probably we're going to have a better offspring. But we know that also we have some shared genes with our grandfather, grandmother, and cousin. So then that's why we built up a pedigree. So then during this pedigree, with this simple concept that how many percentage of our genes are shared, we make a pedigree. This is one concept in the starting point of the genetic and genomic evaluation. The other concept is a trait. So then basically we need to define that which traits do we want to improve it. So then what are the traits? The traits that we say it's a characteristic that we can measure or observe. And we mainly categorize them in two groups of quantitative traits and qualitative traits. Qualitative traits such as having a horn, so that we can observe this one. And this is the qualitative traits. And we see that there's a variation among different breeds, so that we can use this advantage in order to navigate our breeding objectives. Or quantitative traits such as weaning weight. We see that animal at the same age, still there is a range in between. So then we know that there is a variation among them. So then we define traits and we need to have a variation. If we don't have a variation, so then we cannot improve any traits with the genetic selection. But the other concept that it's really crucial, I'm promising that this is the last concept that I'm introducing from the basic concept, but the point is it's a heritability. We know that the performance that we see, it's a function of the genetics and environment. So that means that if we see 30 pounds for the weaning weight, so then we say that this is, say, for example, 12 pounds is because of the genetic potential and 18 pounds because of the environment. So then the proportion of these variation that we see in population, it's coming from genetics we call heritability. So then for the weaning weight, it's 30%. So that means that the variation in weaning weight that we see, 30% is because of the genetic potential. And if we have this heritability number, higher number, say 50, 60, means that we can improve rapidly with genetic selection. And in a general rule, for reproductive performance traits, heritability is low. It's about less than 10%. So that means that if we see that we have a lower reproductive performance, so that at that time we need to improve our environment. But for carcass traits, we can exactly and precisely and rapidly we can improve with the genetic selection. So then this is the other concept to better understand the heritability, basically how much of that characteristics or trait a lamb received from its parent. Say if animal has a foreleg, for 100%, their offspring going to have 4 legs. So then that having a foreleg is 100% heritable. So that for the dog score, it's a 20%. For the weaning weight, as I talked, it's a 30%. And for carcass trait and the wool quality traits, we have a really high heritability. With these 3 concepts, pedigree, variation, and heritability, beef industry has done a really, really great job. This is an example from Angus Association. X-axis stands for the year. The data comes from 1980 and goes until 2011. So the 1980 is the base point, and this blue line is for yearling weight. You can see that over 30 years, Angus Association was able to increase yearling weight for 90 pounds. And this is for mature weight. As you can see in the first 10 years, they thought that it's good to increase the mature weight, but later on they said, oh, no, we need to kind of to make constant for the mature weight. So then you can see here after 1990s, they come to have the constant trend for mature weight. And the green line is for birth weight. And you can see that they start to increase until 1991. Then later on, they found that with increasing birth weight, they're going to have calving difficulty. So then they start to work to decrease the birth weight. So then this graph shows that with the better understanding the pedigree concept and writing and recording the pedigree and recording the traits and having a variation for that traits, you can navigate your objective in any direction that you would like. So then by now, if you have any question, either use chat box, or if you would like, you can mute yourself and ask question, then I will continue. Any question? Okay. Hopefully it's clear that nobody has a question. In case if you have any question, just also you can just hang on and then you can ask at the end of the presentation of it. So I have given example of the Angus Association because we know that purebred association, they keep track of pedigree. But the question comes here, what happens if we don't have pedigree information, or in a commercial setting, we use multi-sire. Basically, we have one sire with 20, 30 dams, and maybe just for 200 ewes, we have, say, 4 sire. Then how do we know that who is the father? So then we cannot build up the pedigree. So then we cannot take advantage of genomic information. But with the advancement in the genomic information, there's a solution for that one, that beef industry, particularly commercial producer, they are taking advantage. So then at that time, genomic information requires DNA. So then where we can find DNA, any cells, but for convenient way is to taking a blood sample or hair sample from animal. So then what is the DNA? DNA stands for desoxyribonolic acid. We don't care about that abbreviation, but DNA has an instruction to create an organism. So think about a cookbook with a different recipes. That cookbook is a DNA, and those recipes are genes. So then those genes basically dictate the body what sort of the trait they should have. For example, having a particular color of eye or particular color of hair, it's dictated by that recipe. So then, but the DNA is made of kind of, for the scientist people, it's made of the different CATG, and it's kind of the 4 block we have for DNA. Kind of consider like these 4 different color, but with different 4 colors or alphabet, you can make any different characteristics. So then there's a huge variation. By far, there's a 20,000 genes that discovered for the sheep. So then we use DNA to do the parentage assignment. As you responded earlier, we know that we share 50% of our genes with our dad. So then in a lab, we determine based on the DNA sample for the particular lab, who would be the potential sire. So then we can build up the pedigree. And we call that one parentage assignment. And we use those information in order to guess the genetic potential for each animal. This is an example that I brought here from Beef Booster data. So then each line is for each sire. So then in the first block, we have the bull identity. What is the bull ID and the birth date? So then later on in the second block, we have expected breeding value. In other words, what is the genetic potential for each bulls? So then you can see that we have a different number for the first bull. We have a birth weight of 3.60 pound. For the second bull, we have 11.6 pound. So then for each trait, we have different expected breeding values. And here are the third column is the actual measurement. So then when a producer comes to buy a bull from Beef Booster, basically they go over these data, and based on their herd concern, they select the bull that's more appropriate for their breeding decision. I'm in this next slide, I'm going to deep dive in a little bit more in these concepts. So that, for example, you go and purchase a bull, and Beef Boosters or any other organization, they give these data, and you see that there are 2 sires. newbie and fluffy, and you check their numbers and you see you would like to purchase a bull to improve birth weight and weaning weight of your herd. So then basically this -2 means for newbie means that we expect offspring of newbie have 2 pounds lighter birth weight and on average. And also we say that offspring of newbie, we expect that have 5 pounds higher weaning weight compared to the average of the herd, giving that environment condition is constant. So then if this is the comparing these two sides, if you select newbie compared to fluffy, you are going to have 7 pounds lighter progeny on average at birth weight and 7 pounds lighter weaning weight. So then you can see like that if a sire is going to be give a progeny of say 20 lambs, so then you can multiply this number and you can see that how much make differences just selecting an appropriate sire. So then from my perspective, this is a really good opportunity and starting point for Alberta lamb producer, just thinking of building pedigree and starting doing the trait measurements. So then by doing so, this is the first step, then you can take huge advantage of the genetics and genomics. But this is not the only concept for the genetics and genomics, but this is a starting point. And then, so then basically the question is, if you have pedigree and you see the variation, and if you record that traits, is it enough or you can do more with the genomic information? And we see right now that Angus Association, they are heavily invested in the genomic information. Then what are the advantage of the genomic information besides building the pedigree? So that, as I said before, there are some traits that they are low heritable, such as reproductive performance or fitness trait. So then basically with the genomic information, you can also improve those traits. Or there are some traits that they are difficult to measure, such as feed efficiency. So that with the genomic information, you can improve those traits as well. But there are some traits you need to measure when they're animals later in their life. For example, carcass traits or longevity. But with the genomic technology, when you take a blood sample right at the birth, you can determine that particular animal's genetic potential. And right at the time, you can decide that whether you want to keep this animal in your herd or you want to cull it. And also, as you recall, I said that expected. This is kind of the guess action, but we do our best as a geneticist to give more accurate estimation. But with including genomic information, basically we can increase the accuracy of our prediction. So then, and sometimes there are simple traits as being horn or pull. So then just with the one gene we can determine. Or so here you can see like the variation is really important, and the differences between the tallest person in the world and the smallest person in the world is because just about one genes. So that again, you can see like the myostatin gene in beef cattle, it makes a really different in appearance and the carcass yield. So then with that, having an understanding genomic concept, so then you can just select for any of those traits that you would like. So that, as I said, you can use for simple trait, or you can use for complex trait or economically important traits such as weaning weight, and you can determine the animal potential right at the birth for any traits that you would like to direct your selection. So that, because we know that sometimes genomic information, getting genomic information is expensive because this is something to do with the lab, so that they design different panels for different purpose. And of course, it gives a different accuracy, so that if we have a higher panel, which is expensive, we can get more accurate data. But for some purpose, for example, for the parentage assignment, it's just you can use 140 SNPs. And for the beef industry, it costs each sample $7. But for the different purpose, as I said, you want to determine animal genetic potential for weaning weight. Probably the 6,000 SNP, which is about $15 per sample, it's a good enough and good enough markers. So then another example from beef industry in Alberta, and it's the first in the North American, our group, we developed a kit we called NVigor HX, NVigor kit. Basically, we know that in a commercial setting, we just keep crossbreeding different breeds. And so we don't keep track. And sometimes we don't take advantage of crossbreeding. So these NVigor, we can use it and we can determine breed composition. It's kind of the ancestor.com, if you have heard about that. You can just use sample and define the breed composition. So then at the end, also you can get recommendation that what would be the appropriate sire breed composition in order to add to your herd. And we show that with the 10% increase in Vigour score, you can increase weaning weight, and you can decrease first calving or and pregnancy rate and weaning rate. So that it's cost about $45, but it's a package comes together. You can build up the pedigree, you can better understand breed composition of your herd, and they can determine also genetic potential for some of the traits that you may be interested. So then this is the end of the presentation. And the take-home message that I would say is, I expect that you had a better idea about the importance of the building pedigree and why do we use the pedigree, and sometimes that we are not able to build the pedigree, how we can get help from having a genomic information. And also there are some examples that I provided, hopefully kind of to help you to have a better idea about the application of genomic information. So then for you, you can think about that if you want to implement genomic information, like how can I make genetics or genomic selection? And the other point is, with the genomic and genetic selection, you are going to kind of have a permanent and cumulative progress in your herd. Basically, the trait that you are selecting for this next year also, you are adding up in this genetic potential. But the point is, you need to be clear. What is your objective? Do you want to improve weaning weight, or you want to improve carcass weight? These are a different question that you need to think about that. And if there is a variation among animal for that particular trait, you can improve. As I said, weaning weight, you see that the animal at the same age, same breed, still there's a variation. So then you can improve with the genetic selection. So then, being said, I would be more than happy to answer any question that if you have. And thank you so much for your time and attending this webinar. Ashley, back to you. And
thank you, Ghader. I'm just waiting to see if there's any questions coming in from the participants. To the participants, if you would like, you don't necessarily need to type a chat box question. You can just simply unmute yourself and ask the question. We'll wait about another 30 seconds or so to see if there's any questions that come through.
sure.
I can kick this off, Ashley, if you don't mind.
Sure.
It's Ali. Ghader, thank you very much for your presentation today. It's great to see you after a while. So thank you for making time for the sheep industry. I was wondering if you could comment a little bit about the importance of data collection. One of the things, having worked with the sheep industry for the last couple of years, one of the challenges that we have, or an area that could contribute substantially, to my knowledge, to the improvement and advancement of the industry, is proper data collection. Can you make a comparison as to what are the basics of data collection? What are the routine data that, say, the beef industry or the dairy industry collect on a regular basis, and how that would make an impact?
Yeah, that's a great question, Ali. Yes, basically we have a simple word in genetic analysis. We say that garbage in, garbage out. So then that's a bottom line for data collection importance. So that means that if we don't have a really good data, so then we don't expect good achievement. That's a main point. But from the which traits that dairy or beef industry they're collecting, depend on their breeding objective. For example, dairy industry, they are more on recording milk production traits. But the other side, in the beef industry, they would like to have more carcass, so then the better carcass quality. So then, but the point is, beef industry is compromised from 3 different sectors, which is calf operation, feedlot yard, and the retailers. So then the point is, calf operator, they don't get premium for the carcass trade. So then, but they get premium for the winning weight. So then they mostly take care about a birth weight and winning weight. So that these are the at least 2 traits that they measure. But later on, sometimes they get some pressure from feedlot yard. So then at that time, they also kind of have a little bit lean toward to making, including some carcass trait in their decision making. But mainly for the starting point, it's a birth weight and winning weight and yearling weight. These are 3 main traits that's a good for starting point of view. And the same thing for sheep industry, I would recommend to collect data on birth weight, winning weight, and yearling weight. And as I said, this is a good data quality comes with a good skill. So then they can have some sort of the good skill to start to do the measurement. And the other point is the building up the pedigree. Having just a damn information works, but it's incomplete, because as we said that we get 50% of our genes from our mother side. So that's the same case. But in order to have a more accurate estimation and have a good progress, basically they need to build up pedigree, means that having both sire and dam, which is, as I said, it's a $7 per sample for the beef industry, but I'm not sure how much would cost for sheep industry. Yeah.
Perfect. Thank you very much.
No worries, yeah.
Thank you for the question, Ali. Are there any other questions from participants today? Since I don't hear any other questions coming through, I'd like to thank you, Ghader, and to all the participants who joined us this morning. This will conclude the webinar. I will be sending an email to everyone who registered for this morning's session that will include a link to where you can access the webinar recording.
Yeah. Thank you so much, Ashley, and thank you everyone for attending today's webinar. Hope you enjoy it.